except you cannot have any devices. You can bring in everything. Yep.
So we're not limited to one page anymore?
Not anymore. This morning, I'm like, that is really hard. I'm like, oh my God, that is hard. And I cut it. I mean, I had it, I took a lot of stuff out. So you can use anything you want. You cannot have a device. You can bring your whole binder in. Now, Kimmy, you do a beautiful job of putting things in your computer. You can't use your computer. But you can print it. Okay? Anything you want. So I don't care if you have a stack of notes this tall, you can use them. Now I'm gonna warn you that if you try to rely on your notes for everything, you're not gonna finish. It is long. There's 30 multiple choice. There is about 10 that deals with characteristics of how we identify organisms. There is about 15 of them that deal with the pathogens of the viral and the prionic stuff we talked about and the fungus. I didn't include any parasites at all in any of the matching. The names.
How many short answers?
Huh?
How many short answers?
One more time.
How many short answers?
9 or 10,
Okay.
all right? But there's also a page of fill in the blank,
Okay.
okay? And we'll do a couple of them here so we can
Why did
practice.
you include any parasites?
Because somebody said there were too many, and I don't remember who said that, but I took that, when you say something, I remember it.
They're on the exam, they're just not in the multiple.
They're just not in the matching part.
Oh, okay.
Right? I mean, I'm gonna ask about the cyst and the troph and all that, but it won't be a lot about the individual names. There's a few, but not very many, okay? So let's do the review. Thank you, Myron, for setting me straight, because I would've kept talking. What is responsible for checking nucleic acids in viruses?
A.
No, not A.
B.
C, Donna says this, that's right, C, the capsid. Okay? What allows intestinal parasites to enter the body? Protozoal. Protozoal. Which one?
A.
No, think you're playing. What does this mean, claw?
C.
Oh, E, okay, E, that's right, the cyst. Lichens are symbiotic relationships between?
D.
Okay, D, yeah. A fungus and an algae, that's correct. Which of the following is associated with scabies? So these are the types, and I didn't ask very many, Kimmy, but these are the types I asked about the parasites.
C.
Not B,
C.
C is correct. Sarcop, did you say C?
I said C.
My bad, I can't, I'm not hearing well today. Which of the following groups of animal parasites have segments called proglottids?
C.
C, correct. What is cyst in penetrating cells and what class does that protozoan belong to? So it's apical complex, so what's the class? Apical myplexa, C. Okay, here's an example of what I meant by characteristics that we define, that define the organism, how we identify them. So on the right side is a type of infectious agent, on the left is a characteristic. So motility, what is characterized by motility? Protozoans, C. How about segments?
D.
D in metazoans, that's correct. What about mycelia?
B.
B, fungi. What about ova? Metazoans.
D.
D. Naked or enveloped?
Virus.
Viruses, A, B. Asexual spore formation? Fungi. Number of legs? Exoparasites. Type of nucleic acid associated with the infectious agent? Viruses. Pigment stored?
Algae.
Huh?
AC.
Algae. Which one is algae?
Dinoflagellate.
The dinoflagellates. AC, that's right. Type of cell wall? Bacteria. Right? Something like that you're gonna have. And then there's gonna be some fill in the blank, right? The something is a stage of development in protozoal life cycle that enters the body typically through the fecal oral route. And that is, no, not the troph. The troph causes the infection, but it gets in by the cyst. Very good. Something are the only group of metazoans that are considered true worms.
Nematodes.
Roundworms is the common name for what I'm looking for.
Nematode.
Nematode. The is the organism known to carry Lyme disease, Borrelia burgdorferi, in the saliva. Huh? No, not the genus, just real common. I mean, you could say amblyoma or exodes, but I'd rather you just tell me it's a
Tick.
tick. Okay?
Lyme disease, take that.
All right. Describe the most important parts of the life cycle of protozoal and metazoal parasites. So for protozoa, what are the two most important parts of the life cycle? Cyst and the, not the ova, cyst and the trophozoite. Tell me something significant about the cyst.
It's non-modal.
Okay, that's an important, but it's not significant when it comes to, it's protective. It's the way it gets into the body. Now, tell me something important about the trophozoite. Now you can say about the fact that it swims. That's really important for it, but it also causes the infection. Let's go to the metazoa. Ova is a sexual thing, right? It causes variation, but it's also mass produced, right? As part of the way that it's disseminated. It's the way it gets into the body. And what about the adult of the metazoal parasite? It's what causes the infection or the infestation. Okay? I had some things about the life cycle of malaria. I took it out. It was too much. I really studied. I'm sorry, but there's still questions about the life cycle. It's just not, you're not gonna have to draw the life cycle.
Okay.
Okay? I mean, I had to cut it down a little bit. Briefly explain the purpose of the lytic and the lysogenic cycles in bacteriophages.
Lysogenic could be non-symptomatic.
Good, and what happens in the lysogenic? That's correct, Christine.
So when it gets stressed and
It goes dormant, and all the cells that come from that cell have the virus inside of it. Okay? And the lytic cycle is what? The replication cycle. The replication cycle, and it's what causes all the symptomology. Okay? That's perfect right there. That's all I need. I don't need anything more. You don't have to talk about the steps. You don't have to talk about attachment and penetration and you don't have to do that. There's other questions about that. Differentiate 3 species of mosquitoes in Texas by morphology and infections transmitted. So there are 3. Go.
Aedes.
Aedes, stripes on the
On the legs.
legs, very good. And what infection does it transmit?
Zika.
Zika's good, or dengue fever.
Not
Chikungunya.
Yes,
That's
it's
kind
like
of
chicken.
that's kind of the hard one to say, right? So I would stick with probably the easiest one is dengue fever. Okay, the next one is what?
Anopheles.
And it's what?
Very big.
It's rotund, and it transmits malaria. An easy one to remember. And then the other one is the small dude. It's called Culex, and it transmits West Nile. Perfect. It's gonna be there. I like that question. Okay? And there they are. Yeah. Okay, so there are 2 case studies. One of them is protozoal, one of them is fungal, right? I couldn't put them on here because then you would have the answer, and I want you to see how well you think about things, because now you're gonna have to use a lot of skills to develop answers for these things. Most of the answers are in your notes or they're in the description. So I couldn't give you the one that I'm gonna use tomorrow, because then you don't get any challenge. But this is an example of one. This is a very easy one. I'm gonna tell you up front it's E. coli, right? So let's just talk about it now. So I'm gonna give you a case study like this one, right? So the patient was a 19-year-old female with a history of urinary tract infections, UTIs, 4 months prior to admission, for which she was treated with oral ampicillin without complication. 5 days prior to this admission, she noted she'd been getting nausea without vomiting. 1 day later, she developed left flank pain, fevers, chills, and noted urinary increase. She noticed foul-smelling urine. Will you see if there's somebody out there? If there's another class out there? Okay. So, where was I? She presented with temperature of 101.8 Fahrenheit. Physical examination showed left costal-vertebral angle tenderness. Does anybody know what that is? Stand up, Suzanne.
Costa would be
Stand up.
red.
It's right here. Okay? So what does that say? Kidney. Right. So, urinalysis with a clean catch urine sample was notable for greater than 50 white blood cells per high powered field. Those are the ones that engulf the infectious agent, right? 3 to 10 red blood cells per high powered field. That means that there's damage being done, right? And 3+ bacteria. Urine culture was subsequently positive for greater than 100,000 CFUs of a gram-negative lactose fermenting rod per milliliter. This bacterium was beta hemolytic on sheep blood agar. So first of all, just sit there for a moment and reflect on how much of this you already understand, because you've taken this course. And we ain't even got to the good stuff yet. Developing the good stuff. Still to come. So what's the etiology of this infection? Well, you have beta hemolytic organism there. You have, this is a McConkey plate. This is gram-negative. It's McConkey. It's, what is it, lactose positive, lactose negative? Lactose positive. And this precipitation that's in the actual media is called what? Bile precipitate. So we know if it's bile precipitate, Kimmy, that it could either be E. coli or Citrobacter. Well, I've already given you the answer. It's E. coli. But this validates that, right? So you could talk about that. What route does the microorganism use to get into the body? This is, of course, in the urinary tract. It's in the kidneys, so how to get in? Through the urethra, right? And what could the patient have done to minimize the opportunity to contract the infection? Not really a fair question, because you don't know any other activities that they were doing, right? I could have said something like, that this was a young lady, 19, who just recently had a new boyfriend. Right. I could have said that, and then that would have led you to believe what? They need to be a little bit more careful about whatever they've done. So that's not really fair, but the idea is there because the questions I've asked on the exam are fair. Okay? I'm looking at your thought process. You may get the wrong answer, but how you came to that answer is what I'm interested in. Okay? I don't care if you get it right. I care about how you think about it. Okay? Maybe 2 of those. Was that helpful?
Yes.
So now let's take about 10 minutes and let's go to lab, because there's a lot to do in lab.